I'm Savannah Knott, and my RCEU summer project was kidney tissue preparation for 1H-NMR analysis and colorimetric activity assay of mitochondria. Metabolomics is the analysis of metabolites in a biological specimen. We were studying the metabolites present in kidneys of LU1WR1 rats, and that helps us understand how the metabolism works and how the concentrations of the metabolites were affected by the diet that the rats were fed. NMR Metabolomics is useful because the NMR is a non-destructive type of spectroscopy and it's easily quantifiable. It's also the most information-rich type of spectroscopy. So the purpose of this study was to assess the activity of mitochondria and identify and quantify the metabolites present in the kidneys of LU1WR1 rats. So those rats were used because they're genetically predisposed to diabetes, and we were studying their kidneys to see the amount and what metabolites were found in them based on the diet that they were fed. So half were fed a control diet and half were fed the moderate sucrose diet, and we needed to see that if the moderate sucrose diet changed the amount of metabolites that were found in the kidneys or if they affected the mitochondrial function. So we needed to cut up the kidneys into smaller pieces and use hydrochloric acid to denature any proteins because we were only interested in the small molecules, and then homogenizing it to get it down to liquid, and then centrifuge it and take the pellet and then centrifuge the pellet again and lyophilize the supernatant, which you need to use later for the NMR. So as I mentioned, we separated the pellet and then we resuspended it and created another pellet from centrifuging it again. And we did that so we could see if there was a difference in the mitochondrial activity between pellet 1 and pellet 2. So we think like the mitochondria gets spun down to pellet 2 and pellet 1's just like bigger pieces of cells and stuff. And so we ended up setting up the reactions with both pellets, but pellet 1 was set aside because we determined that there was no mitochondrial activity really in pellet 1. So the mitochondria activity was measured using a succinic acid assay, and we preheated the fluorescence reader, and then we also preheated the plate before we added the mitochondria at 37 degrees Celsius. So we set up control wells and reaction wells, and you had to add the mitochondria right as you were going to run the reaction because it moves very fast. We measured them at 30-second intervals for 3 minutes at 420 nanometers. So to obtain your spectrum on the NMR, you have to use your lyophilized sample and reconstitute it, and be sure to add DSS and D2O because that's what we had our lock set on. And then once you put it in the NMR tube, you just put it into the NMR and make sure all your settings are what you want them to be. So for this instance, we use 1H-NMR presat CPMG, and we used 128 scans and an acquisition time of 2 seconds and a relaxation delay of 5 seconds. So that was just our settings to get the spectrum. And then once you get the spectrum, you can use Mestranova to kind of fine-tune it and do all your quantifying and to identify the peaks, so you can see from the spectrum below that you can name the peaks and set the widths and everything like you need. So there was no significant difference between the mitochondrial activity of the two diets, but there were some outliers, and We also think that if we had have done the activity assay differently, that we might have gotten a more conclusive result, because there was a difference, just not a statistical difference. There was also no significant difference between the concentration of alpha glucose in the control diet and the experimental diet. And there was no significant difference between the concentration of beta glucose in the control diet and the experimental diet. There was no significant difference between the amount of alanine in the kidneys of the control diet versus the experimental diet, but we did have that one outlier. And there was also no significant difference between the amount of lactic acid in the control diet kidneys and the experimental diet kidneys. So as I mentioned before, we tried several different protocols over the course of the summer, and those results looked promising at first, but then later on we realized that there were some things that we could have changed, but once you start, you can't necessarily start over. So for future studies, we would definitely change the activity assay when you add either the succinic acid or the ferrocyanide or the mitochondria first. It changes how the reaction occurs and at what rate, and also warming them for different amounts of time in the plate reader before running the reaction changed things. So it could be the temperature or just the order in which we added the things, or it may just need a different assay.